the last and final step which is going to terminate this process of protein synthesis is termination. Okay. Now this is step 4, it involves termination and it is going to involve terminal tRNA. Now what would be terminal tRNA? It would be the last tRNA which will have an amino acid joined to it. Now you remember when we did the code dictionary from the genetic code, we had said that there are 3 codons which are known as stop codons, right? 3 of them were called as stop codons. Now why are they called stop codons? Because at this site or at this sequence of mRNA, the translation stops. It comes to a standstill because these codons or these sequences on mRNA do not have any corresponding tRNA. No arrangement of tRNA is possible where the anticodon loop would be having an arrangement corresponding to these sequences. Or simply you can say that these 3 codons do not have any tRNA for the corresponding, do not have any corresponding tRNA. Now supposedly you have this, again same thing, this is mRNA and we have a smaller subunit, we have a larger subunit, we have the terminal tRNA over here in the P site and a long polypeptide chain has been formed by the amino acids. Okay. The formation of peptide bonds have taken place. 1, 2, 3, 4, 5, 6, 7, 8, 9. You can imagine it in hundreds and thousands. Now what happens is this terminal tRNA, let it be, this is having the last amino acid. It comes and joins. Now over here either of the 3 sequence lies, UAA or UAG or UGA, any of the stop codons lies. When the stop codon is identified, what happens is over here comes the release factor. This is being read and release factor comes into scenario. First of all, this A site stays empty because there is no tRNA. As it stays empty, this is the terminal sequence. What this release factor does is, it breaks the bond between tRNA and the last amino acid. The terminal tRNA's bond with its amino acid at the 3' end is broken down by the release factor. Okay, this release factor is GTP dependent. Okay, GTP dependent release factor comes into the scenario, it breaks down this bond and now this tRNA goes from here. What you are left with is a polypeptide chain and that is what you wanted. You wanted a polypeptide in the end of translation. So we get to know that the translation comes to a halt in the stage of termination. Now this stage is possible when we have a stop codon in the RNA sequence. Okay, the RNA, when it is having which RNA? mRNA. When on mRNA there lies a stop codon, then whatever length of polypeptide has been achieved, it would stop over there because there is no tRNA coding for these 3 sequences. When the tRNA does not come over here, A site stays empty, release factor which is GTP dependent, it comes over here and breaks the bond that is formed between the amino acid and terminal tRNA. When the bond is broken, what you are left with is a polypeptide chain and a free tRNA. So this is all that you have to keep in mind. Now I would want you to understand 2 terms before we pile up, that on this mRNA, we had seen that on the mRNA itself, we had seen that polypeptide chain is formed between the sequence where AUG was the start codon and as soon as out of these 3 supposedly UAG came, this much part of the mRNA could form the polypeptide, right? Now further translation will stop until and unless next start codon comes. When the next start codon comes, then only initiation will begin. This entire length of mRNA is not going to give rise to any polypeptide synthesis. So, this region between a stop codon till the next start codon comes is known as untranslated region. Okay. Now, otherwise known as UTR, untranslated region. You have to keep this in mind that between a stop codon and a start codon, the region that is present is known as untranslated region, and the translation stops as soon as the stop codons come on mRNA. Until the next sequence of start codon is achieved, till then there will be no polypeptide formation. So, this is all that you have to keep in mind about protein synthesis. There are 4 stages that we have discussed, and with this we come to the final outcome that the gene has to give. It has to give a polypeptide, and finally we have come to know what happens when the gene is performing its function in metabolism, but how it has carried out at the genetic level, now you are very well aware of. At least I hope so that you are. I have tried my best. I would suggest you to watch these videos all over again, and one more suggestion, that you watch colorful diagrams about these to get a better idea how it happens in real time.